Noswaith dda, a chroeso I chi gyd. Well, welcome everybody and good evening. Thank you so much all for attending tonight. It includes alumni, I know, staff, donors, friends and supporters. Thanks to all of you for joining us today. As you've just heard, the digital showcase this evening is going to be recorded, is being recorded, and we will then send you the link following the event, and you can share that as widely as you like. So I'm Colin Reardon. I'm the President and Vice Chancellor of Cardiff University, and you are here tonight as part of the Cardiff community, comprising graduates who've finished their studies at Cardiff. I'm sure we also have students who are here now, and also friends who are supporting the learning and research of the university. What we're here tonight for is to discover more about Cardiff's research into blood clotting. Blood clotting, of course, is a normal process, and it protects us from excess bleeding, as we all know. But there is a fine balance between too much and too little clotting, and both can lead to serious disease and often death. Now, I would strongly advise you not to take my word for this, but we are very lucky to be joined, and I'm delighted to be able to introduce two colleagues, Professor Val O'Donnell and Professor Peter Collins, and they are real experts in this field. And they're here tonight to demonstrate how Cardiff researchers are working closely with colleagues at the University Hospital of Wales to investigate a range of conditions and make real inroads into diagnosis and treatments. They're both from our School of Medicine. Professor Val O'Donnell is a professor of biochemistry. Her research specialism is in lipids, so you'll find out what lipids are. It's something that Val knows an awful lot about, and their contribution to a range of conditions. And Professor Peter Collins, who is a clinician, and he's actively engaged in with patients in the fight against COVID. He's a professor of haematology, and his research focuses on the diagnosis and treatment of inherited and acquired bleeding disorders. But we'll come first to Val O'Donnell, who will give us the first part of the presentation. Val, thanks.
So thank you very much for the kind introduction. I'm delighted to be here today to be able to showcase the research from my group and also Peter's group. Peter and I have been working together for a number of years. I kind of run the basic science side of the work, and Peter runs the clinical side, and it's really fantastic to work closely alongside Peter to be able to translate our basic science findings into real-world studies in human disease. So as Colin said, blood clots, well, they're a good thing and a bad thing. And the first thing I think I'd like to say is that, you know, it might shock you to know that most of us are actually going to die because of a clot in some form or other. It's a quite shocking thing to consider, but it's absolutely true. Too much clotting happens in many human diseases, and it causes death or serious illness. So, for example, arterial thrombosis, which you'll be very familiar with in terms of heart attack or stroke, will kill 1 in 4 people in the Western world. And then venous thrombosis is associated with a whole multitude of other conditions, such as infection, autoimmune disease, cancer, Alzheimer's disease, where it causes serious illness and death in many cases. And I think as a couple of examples, just to point out, obviously we're all very familiar right now with COVID-19 and the devastating toll that that's taking. Too much coagulation or clotting is proving to be a major problem there. It's something that we'll come back to later on in the talk, but it's really a massive problem leading to admission to ICU and death, and preventing clotting in COVID-19 is a major therapeutic target. Cancer is another one where sometimes the presenting symptom of cancer can be a clot, and sadly people who have cancer and present with clots often have worse outcomes. So understanding the role of clotting in these conditions is really important, so that we can work out how better to prevent them. And then last year there were some studies published showing that a central role of coagulation proteins in Alzheimer's disease for the first time. On the other hand, too little clotting can also cause death and serious illness. There are genetic bleeding disorders such as haemophilia. There are autoimmune bleeding disorders, which often come on later in life, where clotting factors can't work properly anymore. And then there's the big problem of consumptive coagulopathy. This is a situation where there's an underlying pathology or situation that's causing a bit too much clotting. However, the person gets another challenge, which could be an injury or a surgery or indeed childbirth. And then they have a massive problem of too much bleeding, which can't be controlled. And particularly point out consumptive coagulopathy in childbirth, where in the world, in the developing countries, this is shockingly a cause of over 1 in 1,000 live births end in death because of this. Peter is working really actively in this area, and in his talk he's going to come back to a really important trial that he's run, which is really leading to changes in clinical practice in this area. So we will focus on this one later on. So I just want to talk a bit about clots. What are they? How do they work? And what are we doing in my lab to understand the biology of a clot? So I find clots actually quite fascinating, because this is a formation of a clot is something that's really unique in biology. If you consider that you're going from a liquid, a circulating blood, to a solid within minutes, this is a unique event whereby a solid tissue forms in response to a challenge, for example, a trauma injury or an infection. And clots are there to do good things. I mean, if we injure ourselves, we want to stop bleeding. We don't want to let bacteria in. So the clot forms very quickly. It's all of these circulating blood cells, what we call the innate immune hub, coming together to protect us really from the injury that might have occurred. Now, we know a lot about many components in a clot, but what's less understood is how these cells come together, shown here in the slide. All these cells come together, how they communicate with each other, particularly in our case, how lipids are used to communicate as a clot forms, and how they orchestrate or control the production or the generation of the clot. And ultimately, the clot has to stop forming, and it has to resolve and get removed at the end when it's not needed anymore. So in my lab, we try to understand this relatively poorly understood biology of how lipids are used to signal to form a clot. So what are lipids? Well, lipids are another word for fats. And there's a really good example of a lipid that is essential for clot formation that most of you will know about, because it is the target of aspirin. So aspirin is the most globally prescribed drug in the world, and it stops, how it works is it stops platelets from forming a clot by making a lipid called thromboxane A2. So already we know that lipids are essential for a clot to form, and that's just one example. Lipids, of course, also drive inflammation. And on the left of the slide, you can see a typical kind of image of lipids stuck to the inside of a vessel wall, the typical kind of feature of atherosclerosis, which is a vascular inflammatory condition. So lipids drive inflammation and they drive clot formation. And we know a lot about it, but there's an awful lot we still don't know. And in my lab, what we really try to focus on is understanding the lipids, new lipids that we've discovered or we found in our research that drive the clot to form. So just to talk a little bit about that, over the last 10 or 15 years, using a biophysical method called mass spectrometry, we have identified large numbers of lipids made by circulating blood cells. And here's some of the blood cells that make them: neutrophils, platelets, eosinophils, monocytes. We found over 100 different lipid structures. And this is kind of the basic structure. This is one typical example of one of them. And these are lipids called phospholipids that stay in the cell membrane. So I've shown the eosinophil over here. This is the membrane, is the kind of envelope on the outside of the cell. So when the clot is forming, all of these cells make these lipids and they stay in the membrane. And the work that we've done, joint with Peter, and also, using cellular models in the lab, test tube experiments, has really shown very clearly that clotting factors in the circulation require these lipids to form and to stay in the cell membranes, so that the clotting factors can then come sit on the surface, and ultimately they can do what they need to do and form the fibrin mesh that then holds all the cells together to form the clot. So the lipids stay at the surface of the cells after they're made. They help the clot to form. Deficiency of the lipids is associated with too much bleeding. We've shown this a number of times now. And the lipids appear to be generated at higher levels in autoimmune thrombotic disease. So that's kind of where we are now. We're now taking this further forwards with a number of clinical studies with Peter. I'm going to mention briefly a few of them to give you kind of a flavour of the translational work we're doing. So one study, which is joined with Oxford University, with Regent Lee, who is a cardiologist working from Oxford, is looking at the role of these phospholipids in aneurysms. So abdominal aortic aneurysm is a disease which is not that uncommon. Up to 8% of males over 65, less common in females, but not uncommon in older males. It's a silent lesion that appears in your aorta over time, and the problem is that sometimes it's just not caught, it's not noticed unless you have an ultrasound. So if it ruptures, it can be fatal because you just bleed internally. Inflammation, too much clotting, atherosclerosis are all mixed together in these lesions. And when patients do have them and they're being monitored and then they go to get them removed, these patients during surgery are at higher risk of excess bleeding. This is a situation of consumptive coagulopathy like I described earlier. So we discovered last year, 2 years ago, that these phospholipids that we've identified in Cardiff appear to drive aneurysm development through promoting clotting. We published a paper on this, and now we're collaborating more with Regent, and Keith, it was who did the original work in Cardiff when he was working in our group, but we're working more with Regent now to look at a large human cohort of aneurysm patients to try and see what the role of these lipids are, and can we aim to develop preventative treatments from looking at this. Another project in my lab is being led by Majd Proti, who is a Wellcome Trust funded GW4 clinical academic trainee, a cardiologist in training. And he's really interested in how these lipids drive thrombosis in people having heart attacks. So he's writing up his PhD, but one of his main findings was that small lipid membrane structures called extracellular vesicles are playing a major role in the circulation to promote thrombin generation during when a heart attack is happening. Marcela Rosas, who is a postdoc, joined with Peter and I. She switched to look at cells that are deep in the vessel wall. So instead of the circulating blood cells, she was looking at the fibroblasts and the other cells in the vessel wall and asking the question as to whether the lipids on their surface could also be driving coagulation. And she discovered that a protein called tissue factor on the surface of fibroblasts can interact with vesicles to promote clotting also. And then other work, Samia Obaji, who was a PhD student with us, but she's now a consultant hematologist at the University Hospital. She was looking at the other side of this, which is patients with undiagnosed bleeding disorders. So there are many patients who come forward who have problems stopping bleeding. It can get revealed during dental surgery or doing other surgery or during childbirth, and they don't have a diagnosable problem. So we were interested to work with Peter to look and see whether some of our lipids might be missing in these patients. Could that be part of the story? So she discovered that in some of these people with undiagnosed bleeding disorders, that the generation of these procoagulant phospholipids are variable. And so we have an ongoing study now really to look at this in more detail too. Two other PhD students in my group also looking at this in similar things, one in arthritis. It's very well known in arthritis that you have a higher tendency to have thrombotic problems, and the underlying basis of this isn't known. So Daniela is working with Ernest Choi, who's a consultant rheumatologist at Cardiff, to look at patients with arthritis and try and understand the basis of the prothrombotic tendency that they experience. And Ali, who's another PhD student, is really doing much more of the detailed chemistry work and the biochemistry work, mapping the structural diversity of the lipids in human blood. So what about COVID-19? Obviously, this is really on everybody's minds right now, and no doubt there is a devastating involvement of thrombosis in this. It's clearly a major cause of death. It's, again, the combination of too much coagulation mixed in with inflammation driving the coagulation. There's a biomarker now of overactivated clotting, which is predictive of patients ending up in ICU, which is now measured on admission, also a predictor of death. And the sudden deterioration that's often seen in COVID-19 is often down to clots forming in the lung microvessels. And anticoagulants obviously are administered routinely. So in my lab, I'm really interested. Obviously, Peter can talk about the treatment of these patients because he's very involved in treating the thrombotic complications of COVID-19 in Cardiff. But in my lab, I'm really interested to understand how lipids might be involved in contributing to the clotting problems. So I've started a research program to look at the lipid membranes of the virus that causes COVID-19 to see whether lipids we're interested in or other similar lipids, are they on the surface of the virus particle? Could that be contributing to the inflammation and the thrombotic problems that are seen? So in Cardiff, we're analysing the virus particles right now. We're collaborating with Rich Stanton, who is a virologist, Dave Thomas, who's also an oral health specialist. We've got a collaboration ongoing now to look at the lipids in the viral membrane. So on the left, you can see actually an image of these virus particles, and you can see the membrane on the outside. It's very similar, if not identical, to the kind of membranes we see in our own cells. So we're analysing this and looking at the levels and the type of the procoagulant lipids in it, trying to understand the role of this in driving clotting. We're also working with Dave to understand whether actually attacking that lipid membrane could be a potential way to inactivate the virus in the throat. This is not so related to coagulation, but it's very important to think about how attacking lipids could be part of preventing transmission of disease. So that's some ongoing research there. So at this point, I want to say thank you for listening, and I'm going to pass you over to Peter, who's going to continue talking now about a very particular, very specific, really important project that he's been leading on in Cardiff relating to the consumptive coagulopathy of bleeding in childbirth. Thank you.
Okay, well, thank you very much, Val. I'm just going to start the slides. So, as Val says, there is the issue with coagulation is that for most of the time it's there to protect us, but there are problems both when there's too much and too little blood clotting. And I'm going to talk about a situation where there's too little blood clotting, which is associated with bleeding after childbirth. Now, this is a project that has been going on for about 10 or 12 years now, and it's a major collaboration between Cardiff University and the NHS throughout Wales. So bleeding after childbirth is common, but it's usually controlled by natural responses and by routine midwifery care. However, in about 1 in 20 births, there's severe bleeding after childbirth, which would equate to about 1,500 cases a year in Wales. And in about 1 in 150 births, there's potentially life-threatening or life-changing bleeding, equating to about 200 cases in Wales. Now, I say potentially life-threatening because, of course, there's very good treatment available. But in countries where we don't have such a sophisticated healthcare system, bleeding after childbirth is still a major cause of death for women of childbearing age. Now, massive bleeding after childbirth is defined as about losing about 2.5 litres of blood loss. And that's roughly equivalent to about half the blood in your body. And that's the sort of bleeding that we're trying to prevent. Now, massive bleeding after childbirth is increasing. So as you can see here by the red line, and this is data from Scottish audit data, but it's similar across many countries in the world, that massive bleeding after childbirth is increasing. And bleeding of this size has very significant consequences because about 1 in 12 women who have bleeds of this size will need to have a hysterectomy to control the bleeding. And at least half of women will experience significant post-traumatic stress disorder following this sort of experience. Now, bleeding can be worsened by problems with blood clotting, and this is where we became interested in what was happening. Now, this is a picture of a clot, and you can see here that these sort of pink cells, these are the platelets where the clotting starts to form. And the clots are all held together by fibrinogen and fibrin that form this kind of mesh that holds the clot together. Much of the clot is made of red cells, of course, which is why clots are usually red in colour. And the whole thing is activated by a complex system of coagulation factors that end up to generate thrombin. Now, we started our research in this area just by a straightforward clinical observation, and that was that women who were having this very severe life-threatening bleeding after childbirth had very low levels of fibrinogen. So I've shown here the normal level of fibrinogen in the blood, which is about 2 to 4 grams per litre. And you can see in these 6 cases that we wrote up initially, the women during that bleeding had very low levels, much below the level of the normal range. So we started to infuse fibrinogen concentrate and to try and treat this level of bleeding. And what we showed was that we could very rapidly get the fibrinogen level increased, often into the normal range. But importantly, the clinicians treating the bleeding reported a very marked reduction in bleeding at the time of these infusions. And this was the first step on leading us to think that maybe we should look further into the role of fibrinogen in bleeding after childbirth. One of the main problems, however, though, is that if you're trying to measure the fibrinogen level in the standard NHS laboratory, it could easily take an hour to get a result back, and that's too long if you're trying to treat major and severe hemorrhage. We therefore started to look at ways of measuring fibrinogen using point-of-care testing. So this is a machine that we now have in the delivery suite, and it's the size of about a desktop computer. We take the blood, and you can see there that the blood is taken into a tube and just put into a cartridge. The blood is sucked into the machine, and it gives you a readout which is reflective of how good your blood clotting is. Now, the FibTem assay on this machine is a surrogate measure of how much fibrinogen there is in the blood. The wider this trace, the more fibrinogen, and the more narrow the trace, the less fibrinogen. But importantly, we get the result available within about 10 minutes, which means that clinicians can then react to the level and give targeted treatment to correct any abnormalities. So the first study that we did, we asked the question, does a low fibrinogen, or as we were measuring it, a FibTem, if we measure that early during a bleed after childbirth, does that predict progression from moderate to these massive, very severe bleeds? So the answer appeared for there to be yes. So we're looking now at bleeds greater than 2.5 litres. So these are the massive postpartum hemorrhage, which are related to the very severe consequences for the women involved. So if early during the bleed, the fibrinogen is high, then about 6 or 7% of women will progress to these very severe bleeds. However, if early in the bleed, the fibrinogen and FibTem are low, down here below 10, at least half of the women will progress to these very severe bleeds. And looking at other endpoints, we get similar results. So the dark blue here is showing progression to the need for any red cell transfusion. The light blue is needing 4 or more units of red cells. And the green is the needing of an invasive procedure to control the bleeding. So the next question we asked was, if we were to infuse fibrinogen early during the bleed, guided by these bedside tests, would that reduce bleeding and blood transfusion? So very broadly, the outline of this study was that women were recruited early in the bleed and they gave their informed consent to the study at that stage. We did a bedside test of clotting, and if the FibTem was above 15, so to give you some kind of feel for that, that would be equivalent to a fibrinogen of about 3, so well within the normal range of fibrinogen that we would normally see in the blood. So if the FibTem was above 15, we just carried on with routine obstetric treatment. However, if the FibTem was lower than 15 and the bleeding was ongoing, the women were randomized to either receive fibrinogen concentrate or a placebo, and the placebo was just a salt water. Now, importantly, this was a blinded study, so neither the women concerned nor the clinicians looking after the women knew whether or not they'd received a fibrinogen concentrate or placebo. And we looked to see how much further bleeding there was and was there need for blood transfusion. So I don't have time to show you all the details of the outcomes of this study, but this is the key finding, which is that if the fibrinogen was above 2 at the time we gave the treatment, it made no difference to the ongoing bleeding. So whether we gave the fibrinogen concentrate or the placebo, if the fibrinogen was above 2, it made no difference, but below 2, there was more bleeding in the placebo group. So the conclusion we were able to draw from this is that if the fibrinogen is above 2, it is sufficient to control severe bleeding after childbirth, and that early replacement of a low level of fibrinogen is critically important in this situation. Now, these findings, they've been incorporated now into UK and international guidelines, but also they have influenced the treatment of bleeding after childbirth throughout Wales, which is what I'm now going to talk about next. Now, that was done through this project called the Obs Cymru project, and that stands for the Obstetric Bleeding Strategy for Wales. This was a national quality improvement initiative that was based on the research that we'd led from Cardiff, but now brought in the whole of Wales to see whether we could translate that research into clinically important improvements in outcome for women throughout Wales. So what we did was we introduced a package of care to treat bleeding after childbirth that was based on the research that we'd undertaken. We involved all 12 obstetric units, and you can see them on the map where these units are. And importantly, this work was supported by a Welsh Government grant to allow us to translate the research that we'd done to see whether we could incorporate that into routine NHS care. So what was actually done? Well, the first thing that whenever a woman was admitted in labour, a risk assessment was done to assess how much likelihood there was of bleeding. We then started to measure blood loss. Now, this sounds like a very straightforward thing to do, but standard practice is often to just estimate blood loss after childbirth. So we started to accurately measure blood loss, and this is a picture of a simulation where people would go round to all the obstetric units, training midwives in how to measure blood loss after childbirth, so that the accurate amount of bleeding was known wherever you were giving birth. We knew the amount of blood loss. We were then able to use this information to escalate care. So at certain levels of blood loss, we escalated to more senior clinicians to help to try and treat that. In addition, when blood loss reached at 1,000 mils, we did the bedside test of fibrinogen, and if necessary, corrected the fibrinogen level. So these are the outcomes. So I'll just go through this slide a bit more slowly. So at the start of Obs Cymru, that's January to June 2017, what we've done is we've taken that as baseline. And so in the slide, we've normalized that to 0. And then we're looking at the percentage change in 6-month blocks afterwards. So when we look at the number of women who had bleeds of 1,000 mils, then we find there was an increase throughout the Obs Cymru project. And we think that that is due to better recognition of the bleeds, because we're now measuring them and not estimating. Looking at larger bleeds, we saw very little difference in the bleeds of about 1,500 mils or 2,000 mils. But with these very big, potentially life-changing bleeds of 2.5 or 3 litres, we started to see a very significant reduction in the number of bleeds of this size. And so what we were able to do with this project is we were able to prevent the moderate bleeds progressing to massive bleeding. Now, to try and put that into more context for individuals, what it shows is that 54 fewer women in Wales would experience a massive bleed each year. And if we extrapolate that up to the whole UK, that's perhaps around 1,500 fewer massive bleeds. In terms of blood transfusion, at the start of the project, for every 1,000 births in Wales, well, we were transfusing 54 units of red cells. By the end, for every 1,000 births, we were transfusing 40 units of red cells. Again, to translate that into kind of things that you can really think about, during the project, 160 women a year avoided blood transfusion after childbirth. And if we scale that up to the whole UK, that's probably over 4,000 women will avoid a blood transfusion each year. So in conclusion from this work, in most cases of bleeding after childbirth, it's controlled by natural responses or obstetric measures. However, in some cases, blood clotting becomes deranged. The fibrinogen level falls to a critically low level early during bleeding. This causes weak clots and it makes the bleed worsen. What we've shown with our prolonged period of research is that by undertaking bedside testing of fibrinogen and rapidly correcting the fibrinogen, if we integrate that into a package of care, we're able to reduce progression from moderate to massive bleeding. Of course, always there's further research that needs to be done. And the next step is that we need to compare directly the Obs Cymru approach with standard NHS care to see whether the results are applicable widely across the NHS. So just to sum up overall, there is a delicate balance between thrombosis and bleeding. Clotting is crucial to maintaining normal life. It protects us against injury and it also protects against infection. However, many diseases are caused by upsetting the delicate balance between too much and too little clotting. So too much clotting, we've heard from Val about the problem with heart attacks and strokes, deep vein thrombosis, especially in association with cancer, COVID-19, where too much clotting can cause severe problems in people with that infection. Too little clotting, bleeding after childbirth, after a major trauma, in many inherited bleeding disorders, and for diseases such as ruptured aortic aneurysm or after cardiac surgery. Understanding how this balance is upset will help to treat many disorders in the future. And often the problem underlying all of this is an abnormal interaction between the clotting system and inflammation. So thank you very much for listening. And I think we're going to move on now to a period of questioning.
Yeah, thanks very much indeed, Peter and Val, for those really fascinating presentations. And we have had quite a few questions already into the chat. If anybody wants to ask a question, just go to the Q&A and put it in there, and we'll try and come to everybody. If we can't, we can answer them afterwards. So if we don't get to your question, don't worry too much. We will get an answer to you. So there's one which comes up in 2 or 3 of the questions that have come in, and I'll just quote one which is kind of on the same lines. It's from Dana Tate. What could the reasons be for increased incidence of postpartum bleeds? I'm shocked that these trends have been seen in the UK. We've had 1 or 2 on those lines.
Well, I think that the thought is that it's to do with more invasive procedures being undertaken, and often major bleeds can happen on the later pregnancies. If a caesarean section has occurred in earlier pregnancies, it leads to changes in the way the placenta will bind into the uterus, which can increase the risk of bleeding. I think also it has to be said that to some degree it's a better recognition of this. So although the numbers may not be going up as much as it's shown, but it's possible there's a better recognition. And one of the things that we've done by introducing quantitative measurement of blood loss in Wales, the incidence of bleeding appears to go up. But I think that's because we're recognising it better. And so I think there's always that side of things to be considered as well. But there's no doubt that the incidence in many countries is increasing. And that's what really incentivized us to look into this and to try and change the trend. And that's exactly what we've done. We've changed the trend from going upwards now to going downwards.
Yeah, that's a real achievement. So it may be that we're recognising the condition more and therefore counting it more, but clearly there is a rise which you're turning around with, which is fantastic. There's a sort of related question on the low levels of fibrinogen and why. I mean, is there an explanation for that? Why there would be low levels of fibrinogen?
Well, one of the things that happens when there are certain causes of bleeds that will lead to a consumption of fibrinogen. And so the fibrinogen is being consumed into a clot, but it's not an effective clot. So it's like you're using up your ammunition to make a clot that doesn't work very well because the fibrinogen is in the wrong place. And so a low level of fibrinogen is it's the first clotting factor level to fall in postpartum hemorrhage, and all the other clotting factors appear to stay normal until very large bleeds have occurred. So by targeting that specific one and replacing it specifically, we're able to reverse the trend of increased bleeding.
Great, thank you. So I'd like to come to another related question on this, and perhaps then we'll move on to other more general questions on clotting. And there was one which wonders whether lifestyle changes could help improve prospects in terms of this, whether that's lipids in the blood or the propensity to have low levels of fibrinogen, diet in particular, or is this a shot in the dark?
No, definitely. Lifestyle will make a big difference to lipids and your cardiovascular risks. So being overweight and, of course, smoking and the types of food you eat will make a very significant difference. And of course, I think it's been in the news a lot about type 2 diabetes. That's another risk factor for thrombosis. And of course, these are all risk factors for poor outcomes in COVID-19 as well. And so certainly lifestyle issues are very important in these disease processes.
That's helpful, thanks. Well, perhaps we can move on to COVID. I don't know, Val, whether you could say something about what we understand at this stage about the mechanisms of clotting in COVID.
So I think it's really early research going on at the moment, and there's certainly evidence that you know, the inflammation part is likely to be a major part of this. There's a massive inflammatory response to the viral infection, and this will trigger coagulation. There's just no doubt about that. I mean, this causes cells to activate, change their membrane structures, and how the membranes interact with the coagulation factors will be increased. But you know, our research into that, looking at the lipid side of things, is still in its infancy right now. I mean, it's pretty clear that anticoagulants are therapeutically really important now in COVID. So, you know, for example, steroids, which are anti-inflammatory as well. So other anti-inflammatory agents, such as drugs that are used to target cytokines in arthritis, are now seeming to show up very positive findings in trials as well. So the whole interaction of coagulation and inflammation together driving the thrombosis is really going to turn out to be, you know, targeting that's going to be really important in it, and I think that's already happening. So I don't know, Peter, specifically about COVID and the triggers for thrombosis, how much of that's really known yet?
No, well, I think that I think you're right. It's to do with over-activation of inflammation and coagulation interacting together, and I think that we are beginning to start to see some quite interesting information coming through that may link it to other diseases that we already know are related to thrombosis. So, for example, people are just starting to report autoantibodies in the blood that react against phospholipids on the cell membrane. Yeah. Now, these are known for a long time in a disease called antiphospholipid syndrome, and that's sometimes called sticky blood syndrome, and that's known to cause thrombosis. Now, these antibodies are now being found in COVID, and it's quite possible that they're being associated with thrombosis. The question is, why do people with COVID develop those antibodies? And that's something that you might be able to be looking at.
Yeah, so I think some of the work that we've done, which we haven't published yet, was looking at the lipids on the outside of the viral particle, and there's pretty high levels of the lipids that we would classically consider procoagulant on the outside of these viral particles, because these viruses aren't controlling on which side of the membrane they have their different lipids, so they just end up being exposed. And these lipids, interestingly, in other viruses are not just involved in coagulation, but they're involved in viral entry also, so they could become a target for preventing entry of the virus into the cell by blocking their interactions with the surfaces of host cells. So I think it's really interesting to think about the viral envelope membrane composition of procoagulant and pro-inflammatory lipids that may be derived from the host cells that are getting infected, getting inflammatory activated. The virus gets secreted out with host lipids as its membrane, so it's carrying almost messages from the host cell that it's getting secreted from, and we really don't know much about the lipid composition of those viral particles at all. In fact, we're hoping to get some funding to study this right now. We've got a grant in review to really try and look at this in more detail. And I think it's also interesting with the virus too, that there's obviously a lot of discussion in the media in relation to vaccine efficacy changing with mutations of the spike protein. We'd be pretty sure that the viral lipid membrane isn't going to change with mutations, because the viral lipid membrane comes from the host cell. So whatever we do that targets that shouldn't be affected by mutant viruses, hopefully.
So that's a really interesting and valuable line of inquiry, isn't it, on all of this? So we've really only got a couple of minutes left, but there's been one question come up which I think is worth posing, and if we could briefly try to answer it, and that is, given the presentation by Peter on postpartum bleeding and the whole question of COVID, are pregnant women potentially at increased risk from contracting COVID? I
don't think that pregnant women are at increased risk of contracting COVID. I think they have a similar risk to everybody else. I think that what is not quite so clear is what the consequences are, both to the woman and the unborn child. And that's something that's being looked at very carefully. Now, women, when they are pregnant, their coagulation system is set up differently to other people who aren't pregnant. And so how that will interact with the inflammation and the thrombosis that's going on is difficult to know. And again, in Cardiff, along with our NHS colleagues, we are looking at that quite closely at the moment, the changes in coagulation in women who are pregnant compared to women who are pregnant and with COVID. And so we're sort of trying to gather information together, but I'm afraid at the moment don't have any good answers for that.
Thanks very much indeed, Peter. Something I'm sure that people will be interested in, but as yet, it needs more research, as we always need more research. But thank you very much indeed, both of you, Val and Peter, for making the time and sharing your expertise with us. And I do want to thank our webinar audience for being with us and for posing your questions. And I hope you found it as insightful as I did. And as I said, if we didn't get a chance to answer your question on screen now, we will follow up with you directly, so you'll get an answer. Now, we are very proud of the research taking place at Cardiff, and this is just one small but incredibly valuable window on that. If you would like to support us or find out more information, we'll put a link into the comments. And we have a programme of events coming up. Our next event is Rising Stars of Research, which is on the Thursday the 11th of March at 5:15 p.m, which is a chance to hear from 3 up-and-coming PhD students on the research. And I can tell from my own experience that those are always absolutely fascinating, because they're often put onto really interesting bits of research that feed into bigger areas. So you'll get an email in the next few days about that, and also that will have a copy of this recording and links to register for coming events coming along soon. So we hope we will be welcoming you back very soon to another event, and diolch yn fawr a hwyl fawr i chi gyd. Thanks everybody and goodbye.